Darwin observed that living things were variable, and he understood that offspring inherited their traits somehow from their parents. But what he didn't understand was how the information for building bodies got turned into the visible traits that were acted upon by selection. I want to start this lecture by talking about the modern understanding that we have about important biological molecules, particularly DNA and how it works into carrying the information to make proteins. There are four important kinds of biological molecules. All of these molecules are pretty large molecules that are composed of subunits. Now, the way the terminology works here is that individual subunits we call monomers. When they are linked together, they're called polymers. So each of these four biological molecules are the polymers of the monomers that are included behind them. So, for example, carbohydrates you're mostly familiar with from foods. These would be like sugars and starches. Simple sugars are the subunits that make them up. So things like glucose or fructose are relatively small molecules. Something like lactose is a milk sugar, which is a polymer of only two of them. Two simple sugars. But things like starch that's in like potatoes or in other kinds of grains, those are long polymers of a whole mess of simple sugars together. When we ingest carbohydrates, our digestive system breaks down those long chains of starch and turns them into those small simple sugars, which is actually food that you can absorb into your bloodstream and that cells can use. Lipids are another important biological molecule, particularly important because they make up the membranes of cells, but they have other important structural and like energy storage functions as well, and they are polymers of glycerol and fatty acids. Proteins are probably the most diverse set of these biological molecules that we're going to work with, and they are themselves polymers of amino acids. Much more on proteins in a second. And then last, the nucleic acids, things like DNA and all the various forms of RNA, they are polymers of small segments that are called nucleotides. And those, the main function, each of these has like a main function, I guess. Nucleic acids are mainly important for information storage. Carbohydrates are mainly for energy. Lipids are for energy as well as cell membranes. Proteins are really diverse in their function, though. The way that DNA is stored in cells is going to vary a little bit depending on the kind of living thing that we're looking at. Most things that are alive are bacteria, and bacteria have a relatively simple single chromosome that's shown here as sort of this loose coil of DNA that's all kind of wrapped around itself. If you look at mammals or plants or fungi or anything else that's multicellular or any other kind of eukaryote, you'll find that the DNA is divided up into a bunch of little chromosomes. So all of the DNA that is included in an individual's cells is called the genome. Now, so the genome is like the entire complement of DNA. DNA is divided into little segments that are called genes. Now, genes are the recipe for making a single protein or a single like protein fragment. Okay, so the thing I need you to remember for right now is a gene is a recipe for making a kind of protein, and all of the genes that we have that are stored in our DNA, all of our genes that we have, that whole complement is called the genome. The way that DNA is put together, there are some rules. You probably learned these rules before, so I'm not going to belabor it. But the idea is that in order to keep the equal spacing between the two sides of this double helix, there's always some characteristic pairing that we see. So in DNA, the nucleotides thymine always binds with adenine, and adenine always goes with thymine. Similarly, guanine always binds up with cytosine, and the reverse as well. Now, this is important for two reasons. One is this makes it possible for DNA to replicate itself. There's an enzyme called DNA polymerase, which allows the DNA sides to split up from one another. And when those two backbones are split up, then it basically can be used as a template to make more DNA. But this process of replicating DNA, we usually call replication. We can also call it transcription. Proteins are one of the most important kinds of biological molecules, not just because they store energy, which they do, but because they do different kinds of jobs. They're diverse in their structure, and the different structures that they have give them different functions that are going to be usable by cells. I have a few examples here. So they're used for everything from antibodies to be able to fight off, or at least to be able to incapacitate infections. Hormones are chemical messengers that will regulate body processes. Molecules like collagen help to give our bodies structure. Myoglobin and hemoglobin, their names sound relatively similar because they have similar jobs. Hemoglobin, we know, carries oxygen through the bloodstream, and myoglobin is the pigment that makes red meat red. That makes it's also attractive to oxygen. So those two kind of work together in delivering oxygen to the muscles. And one of the most diverse types of proteins are what we call enzymes. Enzymes are molecules that help chemical reactions to happen faster than they ordinarily would at a given temperature. Now, that's important because this doesn't mean that living things are only reliant on the ambient temperature around them for their chemistry to happen. They're able to speed up some of that chemistry through the use of these biological catalysts. And one thing you'll see, I call this an ATPase, which means it breaks apart the energy storage molecule called ATP, adenosine triphosphate. Anything that ends in ACE, you're going to see is some kind of enzyme. There are enzymes that don't end in ACE, but anything that does is an enzyme. So here's what DNA is for. In biology, we have what is called the central dogma. I kind of hate that term only because it makes it sound like when you're dogmatic, it means like you're intransigent to changing your mind. That's not what I want to get across here. We call it the central dogma because this is the central assumption that we have that is borne out by evidence for how things work. Because these are essentially invisible processes. And so I think the biologists and illustrators struggle with how to make the invisible visible. So anyway, just to get to it, the central dogma here is that DNA is read to make RNA, and then that RNA is going to be read to make proteins. There are two different words for reading we use here. So first, the DNA is transcribed by RNA and turned into a simple string called messenger RNA that's a duplicate of one side of the DNA. So that, what's it look like, an avocado? That avocado-shaped RNA polymerase, that's a molecule that splits up that replication fork. And then those loose RNA molecules that are around the DNA are going to be drawn to that area. It sounds kind of weird just to be drawn to it, but at the level of the very small that we're looking at here, it's just like the shapes of these molecules, like the physics of them, that's going to cause them to be drawn to one another. There's no special mechanism for making things join up. They do just that because of the shapes that they have, and the kind of weak forces, the weak interactions between those small molecules causes them to come together. Anyway, that transcript of mRNA is then brought out to wherever the DNA is made. In the case of eukaryotic cells like ours, ones with a nucleus, it's brought out to where the ribosomes are outside of the nucleus. But in bacteria, where they don't have a nucleus, it'll just be brought up to another segment of cell where ribosomes are hanging out. Now, what the ribosomes do is they will read along that mRNA transcript and translate that into a sequence of amino acids. Just like there are loose RNA molecules around the DNA, there are also loose what are called transfer RNA molecules, each of which has a particular set of nucleotides along one end of it, and it also drags with it an amino acid. So the ribosome, as it reads along the mRNA sequence, is going to expose a given section, get those tRNAs to kind of bind up with one another, and then over time accumulate this long string of amino acids. Let me show a little example of how this works. This will be in the learning module as well in case you want to play with it yourself. This is something that's put out by the Concord Consortium, which I think is a simple but correct way to describe what's happening here. So we start here looking at this double helix of DNA, where each of these represents an individual nucleotide, and you can see where it's bound to its complement on the other side. So let's start the model. DNA polymerase splits up the DNA, and then RNAs start to wash in from outside. Now, the only thing to remember with RNA is that though guanine always goes with cytosine, in the case here, you notice wherever there is an adenine, an A, that binds with U, which is uracil. Anyway, so this stuff leaves, in this case, the nucleus, and that mRNA transcript is now going to get clapped together like with the ribosome like a hamburger. And now these transfer RNA molecules, each of which is dragging an amino acid with it, is going to link together these amino acids into a chain. And this continues until it gets to the end of the transcript. There are a few, I should say also, it reads these 3 nucleotides at a time. Each of those 3 nucleotides we call a codon. So at the end here, just notice here, this AUG, that's one of what are called the stop codons. That basically indicates where there's no tRNA that binds with that. So basically the little machinery of the ribosome ends up letting the mRNA loose once it's gotten to the end of the strand there. So then we end up with a bunch of these amino acids that are all bound together. Now that we've seen kind of the movie example of this, let's talk a little bit about the mechanics. So on the right-hand side of the screen, I have an arbitrary string of DNA nucleotides that I've separated into groups of 3. Again, those 3 segments, those groups of 3 segments aren't important to the DNA, but it does have effects on the way that the RNA is going to get read. Now, for a given sequence of DNA, we have a complement of RNA that's going to bind with it. Now, this is one of the things I think is actually really fascinating, is that all living things use this same code over on the left to be able to translate the DNA sequence into... All living things use the same code to be able to translate the RNA sequence into a sequence of amino acids. So what this means is, starting at the beginning, this AUG, the way you read this is if you go to the first position is A, so you go down to that 3rd row, U, that's the first cell in, and then G, that's, if you follow along to the 3rd position thing on the right, it's that very bottom one, that methionine. It basically shows that, as far as I know, at least in the initial transcript, all protein strands on what are called polypeptides, until they get big enough, then we call them proteins. All protein strands start with methionine, to the best of my knowledge. Some of them will be edited later, but certainly in their initial reading, that's how they all start. So this sequence of amino acids comes because the transfer RNA reads this mRNA transcript like 3 nucleotides at a time. So from this, we get this kind of bead on a string structure that I showed in that little simulation, as well as was shown on the right side of this figure. But this is only the very first level of protein structure. This is what we call its primary structure. So the primary structure is only the sequence of amino acids. Now, amino acids are not all the same shape, despite the fact that they're shown like beads here. In real life, the amino acids all have some things all in common. If you look over on the left, you'll notice that, you have a bunch of atoms here, but there's one that's kind of boxed in as an R. If you compare that to the other side of the slide, what it's showing here is that R molecule is kind of boxed off in blue for each of those. That can be as simple as like a single hydrogen ion for glycine, or if you look down to the lower right, it can be those big ring molecules like for phenylalanine or tyrosine or tryptophan, So basically, that's just what we call the variable group. But they all have an amino end and a carboxyl end on one side and the other. So that's what allows them to link together like Legos, but those individual Lego bricks are not all the same shape. So, and the thing that's why this is important is, you notice they have these kind of grouped up into being polar or non-polar or charged, etc. That has effects on how these amino acids will twist the whole strand around. For example, one level up from like the primary sequence, we see that there are both folded over, what are called pleated sheets, beta sheets, as well as these alpha helixes. So this is actually showing not one protein, but 3 different views of one protein, where you can see the twisty helix on one end there. That's just because that comes about as a result of what the shapes of those individual amino acids are. Similarly, the yellow beta strand, or pleated sheet, what's happening over there is that again, that's due to what the sequence of amino acids is there. In between those little gray strings, those are relatively irregular or kind of globular sections of it, so they don't require anything special. But these other views, whether it's the kind of ball and stick version or the full-on rainbow space-filled ones, that's showing what the what the local structure is of that protein. So primary structure is the sequence alone. That sequence determines the sort of local structures. And then the tertiary structure is like the overall 3D structure of a single polypeptide chain. Now, for many proteins, that's as complicated as they get, their 3D structure of one chain. But for some molecules, like the classic example is hemoglobin, there are actually, in this case, in the reds and blues, there are a couple of what are called alpha chains and a couple of beta chains, and so those are going to be those all combine together to make up hemoglobin. So for molecules that have more than one subunit, they have what we call a quaternary structure. Let me show a little example of what this looks like. This is from a bioinformatics website that has a zillion kinds of biological molecules that are put together in it, so that scientists who are interested in making comparisons can share their data. Along the left here, this rainbow color, this is showing, along the left here, this rainbow color is showing where the different parts of the protein are getting encoded on the DNA. The details of this aren't super important, but they relate to the color scheme that you see over on the right. So I'm going to sort of sit this guy turning. So this is a molecule for amylase. Amylase is an enzyme, as you can tell, because it ends in ase. One of the things that it does is it breaks up starches into simple sugars. We make this in our saliva, we make it in our pancreas, we make it in lots of places for being able to digest sugars. Now, some things about this, I'm going to stop the turning. We can change how we visualize this. The rainbow, in this case, like I said, matches up with where along the DNA it's coming from. But one of the things that we can do is actually look at some important aspects of this. So this is an enzyme that's going to be binding with a starch molecule. If we click down here to see the active site, it sort of zooms in, but I'll zoom back out again. If I twist this around, notice there's this one area here where those particular amino acids are kind of more solid, where the rest of them are all kind of grayed out. What this is showing is this is the area that's called the active site. This is where this particular enzyme is going to bond with the starch to be able to break the bond to turn that long chain of simple sugars into just the individual monomers. So this part we would expect to see like relatively conserved across all the living things that would carry this particular enzyme. So anyway, this kind of visualization allows people to be able to compare different structures, see where the action is actually happening, as well as to compare what a given protein looks like across different populations within a species or among different species. So back to the slides. That's the basic function of how DNA works, where DNA is transcribed into messenger RNA. Messenger RNA is then read by ribosomes, which is also made out of RNA, and the amino acids that are drawn along by transfer RNAs get tagged together to form up a single protein. But there are places where genes will change. Now remember, we said a gene is the recipe for making a single protein, or at least a recipe for making a single polypeptide chain. It's possible for us to change what the sequence of DNA is. When we do that, there are different, so these are all called mutations. So mutation is any time you have a change to DNA. Starting up the top here, the most common type, at least the type I tend to think of first, are what are called point mutations, where in the upper strand you start off with a guanine and that turns into a cytosine. These are not entirely random. Some mutations will happen at certain places more frequently than at others, but we can think of them as being basically random. It's also possible for nucleotides to get inserted or deleted from DNA, and that's going to have effect downstream on what the amino acid sequence will be. Let me show you an example. So for example, here there's some point mutations. We can see like two different kinds. So in the first position where I change that cytosine into a guanine, we call that a silent mutation because both GAG as well as GAA both code for the same amino acid, both code for glutamic acid. On the other hand, when we have a mutation in that second place, well, that's what we call a non-silent mutation, where going from TTT to TTA ends up changing the RNA, but now that RNA codon codes for aspartame rather than for what it did before, the lysine. So point mutations may have an effect that will change the structure of the protein, or they might not. So that's the difference between silent and non-silent mutations. Now, if you change the primary structure, does that mean you change the function of the enzyme? Maybe. If you change the structure of the enzyme in a critical place within the active site, then that could make the protein less effective, or it could make it more effective too. But usually, it's more likely that mutations are going to decrease the function of the molecules, the enzymes that they code for. If it's a part of the protein that's in one of these variable regions away from it, it might not make a difference. So there's a lot of contingencies here. If there's a mutation and doesn't make a change, there's no change to the protein, so the protein works the same. If there's a non-silent mutation and you change the amino acid that goes in that spot, that may or may not affect the function of the protein depending on where it's located on that protein. Oh, here's another kind of non-silent protein mutation that's important, where up above I have like an 8 amino acid sequence. Below, if you have a mutation that turns it into one of those stop positions, there's no stop amino acid. That's one of the ways that the mRNA signals the ribosome to stop reading. That could have the effect of truncating this protein. In many cases, they'll truncate it into uselessness, but that will depend again on how far along this happens. If it happens on stop 98 of 100 amino acid protein, it might not be a big deal, but if it stops on amino acid 12 of 100 amino acid protein, then that will be a much bigger problem. Other things that can happen is, as I mentioned before, insertions and deletions. Here in this case, if you delete one of the nucleotides in the DNA, the RNA doesn't know the difference. It just transcribes it the same way as before. But you notice you end up with an uneven, or basically doesn't end it, it's not divisible by 3 sequence of codons there. So you'll end up having these changes in red to what the amino acids are. Frame shift mutations really tend to jack things up because of the way that the code works. I've shown examples of the first 3 of these, but there are other kinds of mutations as well. And again, all of these kinds of mutations because they're any kind of change to the DNA. Gene duplication is actually really important for evolution. We're going to harp on this in a future lecture, but it's also possible for genes to change their position along the DNA strand, chromosomes can get fused together, particularly in plants, it's possible for like the entire genome to be duplicated, which doesn't cause any loss of information, but what it does do is it causes certain kinds of infertilities, and I'll explain how that happens in a future lecture as well. So that's our modern understanding today, but it wasn't always thus. Our modern understanding of genetics, you probably heard the story before, come from the work of Gregor Mendel, who was a Benedictine abbot. So basically he ran a monastery. But in a monastery, people have different jobs. And one of the things that he was interested in, he was a gardener, so he just got interested in how variation in plants was inherited. In a lot of ways, it's really lucky that he picked pea plants to look at, because they have really obvious diagnostic traits, and the particular traits that he looked at were ones that behaved in really predictable ways. Had he picked, cats or something else, it would have actually been a much more complicated problem. So a lot of things kind of converged together on his luck to be able to stumble across a lot of important information that we still use today. So this is a picture of what his little test plot garden looks like today, and also what it looked like in the, say 1860s when he was working. A little bit of terminology here, something you're probably familiar with, want to make sure we're all on the same page about. The information for coding for traits is what we call the genotype. So the genotype is the information, and the phenotype is the stuff that we can actually observe. The phenotype is what is also under selective pressure. You can have a different genetic code, but end up with the same external traits. The environment doesn't care which one you have. The environment only cares about what your phenotype is like. So we'll see that the phenotype is actually what's selected upon. There are a few, you probably learned Mendel's 3 laws from before. And I'm not going to go through Punnett squares and all that in particular, because you guys have done that. But just a couple things to remind you of. First thing that Mendel noticed was this idea of dominance, is that a dominant allele always shows its phenotype. If you have both a dominant and a recessive allele for a given gene, then if you have a dominant allele and a recessive allele together, then it's going to look like the dominant allele. The only time you see the phenotype of the recessive allele is if it's paired up with another recessive. Now, what dominance and recessive means isn't difficult to understand, but the way that I would recommend you think about it is think about it like dosage. Think about it like pigment coloration. A dominant, if you have one allele, one version of a gene that codes for like a dark colored pigment and another one that codes for a light colored pigment, then the darker one's always going to overwrite the lighter one. Now, in this example here, why do the recessive peas have this wrinkled up shape? It has to do with the way that they hold on to water. And so if you have in it enzymes or other molecules that are going to allow those peas to hang on to water better, then they're going to appear smooth. But lacking that, if you have an enzyme that's less effective or less good at being able to hold on to water, that's going to cause a wrinkled appearance. So we tend to think of dominant as better than recessive alleles, but that's not the case across the board. One thing that is generally true, though, is that dominant alleles will kind of overwrite the effects of recessive ones. The couple other things that Mendel put together were that each parent gives different alleles, and those alleles into the sperm or the eggs, and those alleles are separated out from one another. So you call this the law of segregation. And the other thing was that genes, at least the genes that Mendel looked at, sort independently, meaning that what was happening for the wrinkle or smooth part, the surface of the pea, was separate from what was happening with the gene for the color of the pea, or it's separate from what was happening with the height, with the color of the petals of the plant. These basic Mendelian laws were published on, but then kind of lost for a while and rediscovered in the 20th century. And one of the things, speaking before about priority, about the idea of who came up with the idea first, is the one who gets it named for him or her, basically. That's one of the stories here of genetics as well, where German geneticists found, basically independently discovered, dominance and segregation and independent assortment. and I started to publish on it, but didn't realize until later when they found basically Mendel's old book that he'd already done the same work. So what we call Mendelian traits today weren't always called that, but basically it's like this self-correcting mechanism the science has, where you'll give credit where credit is due for whoever generated the information first. And because Mendel did publish on this earlier, he ended up having priority, even though regular university scientists had discovered this later. Mendel did write to Darwin because one of the things that Darwin was kind of hopeless on was how variation was inherited. So Mendel had written to Darwin near the end of Darwin's life, and Mendel had certainly read Darwin. When you look at Mendel's papers, it's pretty clear that he was influenced by stuff that he read in The Origin, but we don't have any evidence that Darwin actually read the letter from Mendel. It came very late in his life, and there's no indication. There's nothing about any of the notebooks, so it doesn't seem like Darwin was aware of what was happening with Mendel. Around the turn of the 20th century, what we saw is actually the sort of eclipse of natural selection as an idea, where geneticists, as more information was coming around, saw the kinds of variation that they could produce as being really important for how evolution could take place. So, this is a picture of the yellow-bellied fruit fly, Drosophila melanogaster, and there are a bunch of little mutations that are shown below. The black and white picture is one of the Cold Spring Harbor, Long Island fly labs. Those are all milk bottles that have cotton in the top and fruit flies in them. And what they would do is the geneticists at that time would try to expose these bugs to different kinds of chemicals or radiation to try to mess with the DNA and then see what the effects were. And by being able to trace these mutations, be able to see how the genes actually seem to work with one another. I'm not going to get into the details here, although it's a really interesting story, but the take-home message I want you to have is that geneticists saw pretty substantial changes in phenotype, which seemed like really important grist for evolution. But they had to do really extraordinary measures in order to mutate the DNA. So they figured that natural selection by itself probably wouldn't work fast enough to be able to explain how we get variation in populations and how that variation is going to get inherited and allow evolution to happen. So Darwin had kind of been eclipsed for some period of time, and sort of the mutationists in the early part of the 20th century had basically thought they had the main explanation for how it is that evolution would take place. Where this ends, and I'm going to tell this story briefly here, where the story ends here is there was a time between the 1940s to 1960s when geneticists and naturalists started interacting with each other more. And there were a series of books and papers that were produced around this time that ended up coalescing around the idea of what we call the modern synthesis, where we're combining the variation of species that was caused by genetic means from mutationists, but then the longer term effects of those mutations within populations from natural selection. So this modern synthesis is basically integrating modern genetic information, modern genetic mechanisms with natural selection to explain how species diversify. Most of the traits that we care about, most of the traits that are important for evolution are not caused by single genes. It takes more than one protein that a gene would code for to be able to cause most kinds of interesting traits for evolution. You're all familiar with simple dominance, and this is what Mendel worked on for the most part. Luckily, because it's a clear story where we're looking at simple dominant, true Mendelian genes. The incomplete dominance I show over here, where again, if we're thinking about dosage, in this case, the red flowers and white flowers, they kind of combine together to make this sort of pink thing. This is the kind of inheritance that Darwin had in mind, and it is true for flower color and other kinds of surface traits, but it's not generally true for most kinds of things. But most of the phenotypes that we care about are things that are actually combinations of a number of different genes all working together. So this super big trihybrid cross Punnett square is showing an example for how skin coloration seems to be inherited. Where if you look along the bottom first at the phenotypes, so there are 6 genes that code for 3 genes in this example that can code for skin color. If a person has no dominant alleles, that 1/64th of the time where they have all recessive copies, you end up with very light skin. If a person has one dominant dose of an allele and then 5 recessive ones, that 6/64ths of the time, then you're going to be a little bit darker. But you notice because like the 2, 3, and 4, there are like a lot of ways to combine those up. A lot of your middling skin tones can come from different genetic ways. So like in different genetic combinations. So the phenotype isn't caused here by a single gene, but by here 3 genes kind of working together. I think the actual number is more like 8 genes for this, but that's not modeled here. What I can show you is here's what it looks like if you have 5 genes kind of interacting with one another. Each of these colors represents different combinations of getting the same phenotype. This gets really complicated, so to the point where most things that are caused by multiple genes don't have a simple Punnett square kind of distribution. They have something that looks more like this. It should be a histogram, but they're showing it as a bar chart, where you end up seeing these normal distributions of, some individuals will be very small, some of them will be very tall, but because there are a lot of different ways to be of an intermediate height in this case, then and that's where most of the population is going to be. So the take-home thing I want you to get from this is that most phenotypes are not caused by the action of single genes. Most of them are caused by multiple genes working together. And by the way, we'll talk more about this later, this ignores the effect of the environment altogether. But even if we're just looking at genes, this is we tend to see normal distributions of phenotypes rather than simple like typological ones with really easy Mendelian ratios. We're going to talk about real natural selection later, but I want to talk about a different kind of phenotypic change that we call just random drift or neutral selection. When we talk about genetic drift, we tend to look within populations to see if a given allele is always present or never present. Now, because genetic drift is random, the example here, the drunkard's walk, is a statistical example, basically to say that you can either you're going to have an allele that's going to be fixed where it's always present, or lost where it's never present within a population. So see, as an example, if you had a bowl of Skittles or something there, and you picked out 2, it's not hard to get two that are the same color, even though they have a 50% ratio within the true population. If you took a bigger sample, your bigger sample is more likely to reflect what that whole population is like. And what I'm going to have you do for the next part of this is to do some simulations. What happens when you have genetic drift, just random change in populations, and then count the number of times that you either have an allele lost or when it's fixed in the population overall. Where we head after this is going to be talking about changes in phenotype that are not just due to random chance, but due to actual selection.